You know, it's a real honor to be involved in this presidential lecture. It's also a challenge because in some ways the stuff that we've all been doing, and you'll see all the people who are involved in this over the last few years, are actually creating a translational story that represents, I think, what the Einstein-Montefiore merger embodies. And so I hope you agree with this, and that's the challenge, is to try to convey that. So what Alan just said was basically dead on. I mean, if you can design a microscope that has high enough resolution that allows you to see events inside of a living animal and take in all of the kinetic events that occur, what comes first, what comes in the middle, what comes last, you can actually understand mechanism of disease progression that's not possible when you look at fixed tissue like a pathologist does. It seems a simple concept, but it's transformative. And so that's what the title is about. How can you use a special kind of microscopy at very high resolution to actually get at the mechanism for cancer dissemination, and in so doing, design new treatment strategies? Okay, so that's the talk. And this is about cancer, of course. And it's about a piece of the cancer puzzle which is particularly vexing. Almost all of our efforts are designed toward dealing with the growth of cancer. So either the growth in the primary site here, number one, or the growth once the cells spread to distant sites at various organ sites. 95% of the funding, almost all of the activity is directed toward the regulation or suppression of growth. But what's really killing the patient is the spread of the tumor, the metastasis, the systemic travel of the tumor cells. And almost no effort is being put to that. And it's not because people are indifferent, it's because the technology doesn't exist. It's almost impossible to figure that out. So what we've been doing over time is to try to develop microscopes that allow us to define these 4 steps that you see pictured here in this cartoon textbook image, and actually look at them in great detail to understand the mechanism of each of the steps, and that way get at the root cause of death, which is due to dissemination. So why use multiphoton microscopes? Because it fits the bill. It actually is the high resolution method of choice. But it's not possible to just go out and be a good physicist and build the perfect microscope and then bring it over to biomedical researchers and have them apply it. Because what you'll get is a microscope that has perfect physics, but actually doesn't solve the biological problem. So when we started the Biophotonic Center many years ago now, and Rob Singer and I direct it, we decided to flip that model on its head. And instead of building the perfect microscope and then looking for an application, what we decided to do was to take biological problems that were otherwise insoluble and then build a microscope specifically designed to address that biological problem. So it has very limited utility, but it is definitive in its use. So we have a triangle now that represents the center with 3 corners. One representing the research of the members of the Biophotonic Center who are listed here. And they bring biological problems and partial solutions. And then we have an innovation laboratory over here on this corner, which builds a microscope which allows them to address their problem. And then as we go around and around and around, new technology comes out of this, which is offered to the Einstein-Montefiore community overall through a core, the analytical imaging facility, that anyone can use. And so the technology spills out to people. And you have to have a very strong, besides the strong science, you have to have a strong engineering, software writing, and mechanics infrastructure. And here are some of the people that are involved on that side. And I'm going to highlight 2 of them in particular, David and Shailesh, as I go along. So the center is important to making this work. And so what does it do? It built this microscope. It doesn't look like a microscope, but in fact it is capable of looking inside solid tissue as if it was transparent as glass at single cell resolution. It's called a multiphoton microscope. And this is actually what it looks like in the flesh. It's about as big as a school bus. And you can't see the laser light because it's in the far infrared. But what it allows you to do, and coupled with windows in animals, and these animals are designed, these animal models are designed so the animals go through their life with the window in it, they live with it. and you can watch the entire progression of their physiology, and eventually in the event of a disease, or we start a tumor, the progression of the tumor and how it spreads. So putting these two things together, the windows and the microscope, you have now an opportunity to watch what's actually going on in the disease. Well, that's great, but now what do you have to make a decision. Do you look at very, very high resolution, so you can see a single cell and inside of a single cell, but you can't, because you're looking at such high magnification, see where the cell is or how it's interacting with the organ system. It's a one or the other. Okay, so what we did is we built a microscope that would do both. And the way you do that is to use large volume imaging. You go in and take a whole bunch of very high magnification images with very high resolution, and you put them together, because if you look at them individually, you still don't have the answer of context. If you take a whole bunch of images randomly, you still can't figure out what's going on. But now if you have a way to put them together in space and time, you now get the phenotype. And that's what you do. For example, here's a breast tumor. This is a 4 cubic millimeter volume of tissue, which now we can zoom in and zoom in from millimeters down to 0.2 microns, and we can watch the green tumor cells entering the red blood vessel in the live animal while it's going about its daily life. Now this is great, right? So now we have solved the problem, only to hit another problem. The next problem is, now you've got terabytes of data, and there is no way you're going to be able to go through it manually to figure out how to analyze it. So now you have to bite the bullet, and you have to go to Aviv Bergman. You have to take a computational systems approach to untangle all of this data. Only then will you have a hope of understanding it thoroughly so you can get at the biology again. So in this case, what we found was that the Biophotonic Center was inadequate. It was necessary to build an integrated imaging program that would take the combined expertise of all of the multidisciplinary teams in these departments throughout the institution to come to a solution. And so the first solution was to solve this problem, take the information coming out of the microscope design and validation in the Biophotonic Center and put it through the filter that is represented by the integrated imaging program in the form of this very talented mathematician who's the chairman of our Department of Computational Systems. And to solve the storage problem and the high performance problem, we had to bring in a team of IT engineers led by Shayla Shenoy and manned by Eli Sylvester and Brent Soley, who are all in the Office of Research Computing, but of course are working with us to help with the architecture of computing at Einstein, and they turned that expertise on this problem. And so we were then able to analyze the large volumes. And something really profoundly surprising comes out of this. So you take all of these fields of view at multiple depths, so stacks and stacks of optical images in this animal, and you put the whole thing together. You take the next animal, you put them all together, and so on. And then you look at all of this and you say, what is going on in this tumor? What are the prominent tumor phenotypes that are leading to the progression of the disease? And so you put that data through, and out pop the microenvironments where the events are occurring. Then you put the microenvironments back through, and out pop the tumor phenotypes. And you keep iterating this over and over again using a support vector machine vision classifier. And this is music to the ears of Aviv, who's sitting over there. Many of you don't do this, don't understand exactly what I'm doing, but basically you have a robot looking at this data and sorting through it at very high resolution, and out pops a very simple result. So what was discovered? So you have two phenotypes that come out, a fast and a slow phenotype. This slide got jumbled. It should say red slow and blue fast. So here are the fast cells in this 5-dimensional volume plot. Here are the slow cells in red. And you'll notice how they occur relative to different microenvironments. So the slow cells are farther away from blood vessels, have fewer macrophage interactions, and are in relatively sparse collagen. But as they get closer to blood vessels, the phenotype switches to fast, and now the tumor cells are approaching the vessel, the collagen density goes up, the macrophage density goes up. So let's look at the raw images and think about this. So here's the fast, here's the blood vessel, the tumor cells are roaring through it, toward it, as green cells. Isolate these cells computationally, and you realize now that this green cell has with it associated a macrophage, which is the blue cell, and they're literally driving at very high velocity toward this blood vessel here in red. Fast, 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 and then they switch to slow as they approach, and the tumor cell sends out this little finger called an invadopod, which it inserts into the vessel wall and makes a hole. Now, if we reconstruct this volume, remember we have this huge amount of data, we can go back now and reconstruct the volume, place the macrophage and the tumor cell when they're next to each other on the vessel, and then look for the optical section in the data set where we see what they do, and you find that this structure is the point where tumor cells, the green circles, enter the blood. This is the moment of dissemination. And it turns out that this special structure, which we gave the name TMEM for tumor microenvironment of metastasis, is the doorway for this event. So the tumor cells undergo this fast migration. They then transition to slow, where they build the TMEM, and then TMEM is used to inject the tumor cells into the blood. And here's the actual three-dimensional structure at high resolution. There's the invadopod sticking between these two endothelial cells in red, the tumor cell, the macrophage, and the tumor cell's invadopod as drawn here. So this is the doorway for entry of tumor cells into the blood. Why is that important? Because it turns out when you actually watch the thing work, the TMEM work, we're watching it work here, you can see the opening and closing of the door as the release of red marker from the blood vessel. When you now look at what's going on with the green tumor cells, here's the TMEM. You can see green tumor cells entering the blood every time the door opens, and there's a little red flash. The interesting thing about this is that it's coordinated. So the door opens in blue, that's the red, that's the flash, and then the tumor cells go through and the door closes. Now, the thing that's particularly clinically interesting about this is that tumor cells can get into the blood only where there are TMEM. They cannot enter the blood where there are no TMEM. So now TMEM is a definitive marker for metastatic disease. So how does it work? How does it work? It turns out that the macrophage in this TMEM structure is special. It's a pro-angiogenic macrophage that is absolutely engorged with this growth factor, VEGF, which causes blood vessels to become briefly permeable. And you can spot these macrophages now that we know them. We can go in and just look at a tissue section in the pathologist's office that's stained for VEGF. And you can pick out these TMEM macrophages because they're so engorged with VEGF, they light up and they stand out. So every place you see one of these, you have an active TMEM. So what we did to prove the function is we took the VEGF gene out of the macrophage. We literally knocked it out genetically. And then we looked at TMEM function, and we found that the permeability, this event, and the intravasation, this event, completely went to zero. So the VEGF was the key signal that allowed TMEM to do its work. So far we've done very high resolution imaging, we've done very high performance computing and machine vision analysis, and what did we learn? We now could take this relatively information poor cartoon and hang all of the signaling pathways on it and the key structures, all of which now are prognostic for metastatic disease. So we learned that there are only two migratory phenotypes, fast and slow. We know now the signals that these cells use to find the blood vessel and enter it. We identified TMEM. We identified the signaling pathways that make TMEM function, and we got a surprise. That is, we got a method for capturing the disseminating cells at the moment of dissemination. So we can go in and basically make an artificial TMEM, put it into the blood vessel, here you see it, and the cells here that are in red have been flash photoconverted from green to red, and you can see these red cells now enter the artificial TMEM. We can capture them. They're alive. This takes a couple of hours. You now have a population of pure cells that are dissemination competent, and you can expression profile them. And you have a window on what they were genetically thinking, so to speak, at the moment they were disseminating. And what you find is that they're embryonic cells. They're embryonic stem cells. They're tumor cells that think they are day 8 in the embryo making a mammary gland. So the TMEM is actually the cancer's version of mammary gland development. It's a normal process that's been co-opted by the tumor to spread the cells rather than spread the tree of glands in the mammary fat pad. So it's tragic, but these cells not only have the power to migrate and spread, but they have the power to seed whole new tissues, whole new tumors. And so they're very dangerous. Now, the other thing we get from that analysis is the invasion signature. It's the complete wiring diagram that these cells have when they're disseminating. And the wiring diagram looks like this. So here are all of the receptor tyrosine kinases that we discovered by the imaging. They all fire their signals through a switch, a master switch, which is this thing here called MINA-calc. So when you flip from the 11A to the invasive, you turn on the fast and the slow migration, which leads to dissemination. So we have the switch, we have the wiring diagram, we know how it works. So now we can take what was originally a very simple diagram and definitively describe the cell phenotypes, how they interact, their signaling pathways, and all of the markers that identify them in these various stages of function. And here it is. And so the markers that come from this are called MINA-calc, the switch, and TMEM, the doorway structure. And here's how it all fits together. So obviously we're poised now to go forward with prognostics to use on patients so we can decide who is going to have metastatic disease and who is not, who's going to respond to treatment and who is not. So now we have another iteration of the relationship between the Biophotonic Center and the Integrated Imaging Program, where now we have brought in these wonderful, talented antibody and probe developers that you see here. Our machine vision digital pathology team, who are listed here. And of course, our clinical interface, oncology and epidemiology, and we put these 3 bits together, and we get these markers now moved into the clinic. How do you do that? There's a space here, it's my fault. Yulin is missing. Yulin, who's the histopathology person who's been driving a lot of the staining here, I'm going to describe next. I apologize for not having her picture, but she's sitting right over there in the audience. So how this looks when you actually do move these markers in the form of antibodies now into fixed tissue is you get the actual structure, TMEM, that you can see as a pathologist. Here's what it looks like live in the live animal. Here's what TMEM looks like while it's working. And here is how it looks in fixed human breast tumor tissue. Here's the MINA-calc positive tumor cell in red. It's paired macrophage in the TMEM structure, and they're inserted into the blood vessel walls. This is a TMEM. This gets scored. So it turns out that MINA-calc and TMEM are mechanistically linked. So Maya, who's one of the team leaders, Maya Akhtai, is a cytopathologist, so she can go and take a biopsy of these patients. There's 120 patients in this study, and get a score for MINA invasive, get a score for MINA 11A from the FNA biopsy. And then the tissue is scored histologically for TMEM using that test I just showed you. And what you find is that as the MINA invasive level goes up in the patient and the MINA 11A level falls, the switch is thrown toward TMEM assembly through the activation of the fast and slow phenotypes. So for each patient, if they have an elevated MINA invasive, a decreased MINA 11A, they have a high, an ever increasing TMEM count. They're in more and more trouble clinically. So these studies have now been done. You'll notice the authorships. We have Einstein pathologists, epidemiology, pathology, oncology, proving the usefulness of these markers on large patient cohorts. MINA-calc is also predictive. Prognostic for metastatic recurrence. But it's still a series of tests that tend to be used late in the interaction with the patient. The patient's tumor is being resected, biopsies are being taken from the tumor after it's been clinically presented. But what about having something that you could use live early on? So how could we take advantage of, again, this architecture of having the Biophotonic Center interacting with the Integrated Imaging Program, putting the right clinical translational teams together so that we could take the multiphoton imaging and digital pathology, which is informed by it, but take it now into a real-time, live patient MRI test. So to do this, now we have to bring back our digital pathology team, our vet and DVM PhD, who plays a prominent role, as you'll see shortly, in looking at these phenomena, and Craig Branch, who is one of the 3 co-directors of the Integrated Imaging Program. It's Craig, Rob Singer, and me are basically trying to run the Integrated Imaging Program, but we're greatly outclassed by the talent we're surrounded by, but we do our best. So Craig has been the team leader here and has moved this now to what seems to be almost ready for prime time MRI test. So here's how one sees the TMEM activity in 3 different ways in the IIP. One is digital pathology, low, high TMEM count, each circle is a TMEM. The multiphoton imaging, where we can actually see the function live, but only in a mouse, not in a human. We can see this in either a human or a mouse, the pathology, only in a mouse, but now we can take this information and validate the interpretation of this signal in the MRI image as an increase in TMEM activity. So this voxel right here is about the size of this image in the light microscope, but this was done, live in an animal using MRI, not optical imaging. So does it work? Does it work? Okay, so we can see digital pathology. We can see the increase in TMEM. We can see it increasing as a histological structure, as a count. And now when we go from early to late carcinoma, the same thing here, but now by MRI, we can see the TMEM activity increase. And now comparing again digital pathology head to head. Here's the number of TMEM because you can see them histologically. Here's their activity, early versus late carcinoma. And when we inhibit the TMEM by shutting off the MENA-calc switch, function stops. MRI version of that, here it is, early going to late, increase activity, shut off TMEM, activity goes down. So in the not too distant future, we believe that we'll be able to have a patient that will be actually observed during the early stages of treatment by various drugs, and we can assess the effect of the drug on TMEM function and the response of TMEM to the drug. So where are we going to get such drugs? Okay, so how do we inhibit TMEM? Well, when you look at how TMEM works, you get an immediate light bulb goes off. This receptor tyrosine kinase regulates the secretion of VEGF by this macrophage in TMEM. It has many, many inputs, only 2 of which are shown here. So you can't go and inhibit each input, but what you can do is you can inhibit the switch, the receptor tyrosine kinase itself. So we teamed up with a pharmaceutical company who had been toying with switch pocket inhibitors, which are designed specifically to bind to the hinge site in receptor tyrosine kinases rather than the active site, because active sites mutate so quickly that you get drug resistance within a few months. But the switch pocket doesn't mutate. So if you get a drug that jams the switch pocket, you have no drug resistance. And you just have to find the right drug and you're on your way. And we hit the jackpot with ribastanib, which has an extraordinarily high affinity for the switch pocket in the picomolar range. So you feed the animal once a week, you dose them once a week, and they have the benefit of the drug for the whole week, and it's orally administered. You put it in the food for the mouse, or you would have a tablet for the human. In any case, the ribastanib blocks the Tie2, stops the VEGF, and the prediction is that the ribastanib will turn off the function of TMEM as well as knocking out the VEGF gene turned it off. Does it work? Yes. The ribastanib turns off TMEM as well as knocking out the VEGF genes. It's a really good drug. So we did a preclinical trial with a large number of animals that have a very, very aggressive mammary tumor called PY-T. It's an oncogene-driven metastatic tumor. The animals don't live more than a couple of months. They die due to metastatic disease. So in this study, the animals have the tumor is removed, and then they get a course of treatment. If they get no treatment, they die even after removal of the primary tumor due to disseminated sites. If you treat them with chemo, such as paclitaxel or eribulin, they die more quickly. So the chemo is actually, in this particular animal model, is making the tumor more aggressive. This happens in some humans, by the way, but here we see it in this whole animal model because they're all genetically identical. But if we add ribastinib to the chemo, we get a 70% cure rate, which is unheard of for this PY-T. Now you gotta ask yourself, why are we curing the tumor if the primary tumor is already gone? What's still killing the animal and therefore being treated by this drug? There must be tumors out there that are still progressing. So let's look at this. So we know in the primary tumor that this series of events where you go to the fast and then the slow, you build the TMEM, and then you put the tumor cells into the blood. What if something like that is happening at the secondary site? What if there's a secondary site TMEM so that all of these secondary tumors continue to disseminate, and that's what kills the patient, even though the primary tumor is long gone. The surgeon did his job. Okay, so let's look. Now, look at this happy fellow here. So Rob Mitchler, leading our T32 surgery training grant, has supplied some very talented T32 fellows. It's a PGY2 resident who's sitting right behind Alan Spiegel, named Sonia, who actually teamed up with David and built this lung window, which allows the animal to live long periods of time where the lung is exposed optically. So now we can watch the tumor cells arrive spontaneously from the primary tumor and see if the secondary tumor acts the same way as the primary tumor in terms of TMEM dissemination. So this resolution of this window is so extraordinary. You can actually see individual tumor cells in green arriving in the capillaries of the lung and seeding a new tumor here. What's really remarkable about this that you're not aware of is that this is in a live mouse that's breathing at 120 breaths per minute, and there's no optical or computational post-processing here. This is a raw image. This is truly remarkable achievement. So now the key, is there TMEM in the lung? So the pathologists look at both the mouse and the human lung that have these very small mets, and sure enough, histologically, there's TMEM, the 3 cell types. And according to this new window, it's active. You can see it working there. It's squirting out the contents of the blood vessel as tumor cells enter. So TMEM is functional not only at the primary tumor, but it reestablishes itself very rapidly in very, very small mets in the secondary sites and continues on with the disease even after the primary tumor is treated and removed. And this is what kills the patient. So we think our systemic treatment will deal with this because the rules for operation of TMEM are the same at all sites. So this is an opportunity here that we have that we did not think of before this optical technology brought us to this point. So we want to use drugs that treat all sites. Now some of you are asking, and I've gotten this question at this point when I've given this talk before, or this kind of a talk before. So we get that. We get why the animals are dying. There's this tumor progression continuing on using the same rules at distant sites as were used in the primary tumor. But why do the animals that are getting chemo do so poorly compared to the animals that are untreated? And does this ever happen in people? Can your imaging technology tell us anything about that? So here's where George comes in, George and his colleagues. I don't know where his picture went, but it'll reappear here shortly. They looked at this, and they looked at no chemo and chemo, and they looked at whether or not macrophages were coming in, because the macrophages that come into the tumor help build the TMEM. And so just general macrophage populations weren't coming in. But when we looked specifically for the proangiogenic TMEM-like macrophage, the VEGF engorged macrophage, they're coming in large numbers in response, or they're appearing in large numbers in response to chemo in the tumor. and TMM assembles before, after chemo, before, after. This is mouse, this is human, before, after, before. Astounding. So the chemo is actually causing this huge pro-metastatic change. And the amino-calc's going up. The whole sequence of bad events is reoccurring in response to chemo. So the chemo is bringing in these macrophages that induce the tumor cells to throw their amino-calc switch and supplying, in addition, the macrophages that can assemble TMM, so you get a double hit. You get a lot of TMM, and you have a very deadly outcome in these animals when you treat them with chemo. Does this happen in people? Is it possible that this happens in people? So here our clinical team got together and started to look at this in the Montefiore patients. And this red line here in the horizontal position is the transition between low, relatively, low to intermediate risk of distant recurrence TMM score, and above this line is very high risk of distant recurrence TMM score. And we looked at individual patients. They did. So this patient, it got a little bit worse, but you're still okay. Look at this one. Before and after chemo, blue to green. Before, look at this one, before and after chemo. Some of these patients are being significantly harmed by standard neoadjuvant chemotherapy. Nobody expected this. Chemo scores are going up. Some of the patients are fine. What's the difference between these people? We didn't expect this because the studies that have been done looking at the pre and post-operative chemotherapy did not show any difference between the patients. And these studies were published in a variety of very prominent journals. Studies were done in Boston, very believable, New England Journal of Medicine, and so on. So given the data that George was getting, and that Jessica, who's also, by the way, a PGY2 surgery resident, now on the training grant, the T32 training grant, as a fellow, good use of talent, by the way, get him out of the operating room, teamed up with Maya, our team leader, and looked at this in the Montefiore cohort, not the Boston cohort. Turns out that the Boston cohort, and with the help of our oncology and statistician and epidemiologist to make sure we were doing it right, the Boston cohort was 85% white. But then when you look at the Montefiore cohort, the whites do pretty good in response to preoperative chemo. Hispanics, not so good. The blacks, not good at all, really bad. And in fact, here's the comparison. White, mostly white, the black population. It's a train wreck. So this is not a happy outcome. And in fact, is going to be controversial. But the reality is we have the markers where we can follow this during the early stages of chemo and therefore change the treatment of these patients. So these studies now have to be done on large cohorts, and these cohorts are uniquely here at Montefiore. And so this could be a model for a national study. And so I urge the team and the Montefiore community in general to support this. So chemo induces TMM assembly and function. Some patients are much worse off. We think we know, at the beginning, know who they are. And we believe having the markers uniquely here at Einstein-Montefiore will allow us to design clinical trials to define new treatment strategies for patients, depending on what their racial and genetic background is. So individualized treatment. So, okay, where are we? Have you started to do any of this? And the answer is yeah. I mean, actually, there's a clinical trial running, and Joe Sperano is going to tell you about this. There's a clinical trial using ribastanib and chemo combinations, and Joe's going to describe this next. And here's his clinical team, Jesus, Maya, and Joe will give us the details. So before I leave, I just want to mention the people who were not pictured on the slides, who have been instrumental people in my lab over the last couple of years, including these MD PhD students, Max and Edison, and many postdocs, many of who are now gone on to their own labs. And in particular, Program 2 in the Cancer Center, who have collaborated and been extraordinarily good colleagues Ann Bresnik, Diane Cox, Samantha Goswami, Lewis, Richard, Jeffrey Siegel, who we all know, and Vladislav Lakusha. And then I also want to just give a hats off to the histology comparative pathology, where the staining of TMEM is done in the research trials, and then of course the funding. And I want to make a special effort to mention Evelyn Lipper and the Gruss Lipper funding of the Biophotonic Center, and the Lipper Foundation funding of the Integrated Imaging Program, which led to copious amounts of NCI and other NIH and DOD and Susan Komen funding. So it becomes a self-perpetuating grant story. So thank you very much.